Welcome back to The Explainer. So, when we hear the word bacteria in the news, we're almost always being warned about some terrible microscopic villain that's out to get us, right? But what if we completely flip our perspective for a minute? What if we look at bacteria not just as simple enemies, but as brilliant biological survivalists equipped with a truly ingenious playbook? Well, today we are cracking open that exact playbook. We're going to dive into the fascinating and honestly sometimes terrifying mechanics of bacterial genetics and see exactly how these bugs are driving one of the absolute greatest medical challenges of our time.
Here's the roadmap for our explainer today. First, we'll meet the real E. coli. Second, we'll explore the bacterial genetic arsenal. Third, we'll look at the antibiotic resistance crisis. Fourth, we'll cover testing and measuring the threat. And finally, number 5, stewardship and the ticking clock.
All right, let's jump right into Section 1. Meet the real E. coli.
To really grasp this resistance crisis, we first have to clear up a massive misunderstanding about E. coli. It is not some rare, exotic pathogen like the plague. It is literally living inside the bowel of every single human being right now, and it is incredibly efficient. When the conditions are just right, an entire E. coli cell can divide in about 20 to 30 minutes. If you leave it completely unobstructed, a single cell can become a billion organisms in just over a day. A billion. Let that sink in for a second. That astonishing speed, that's the ticking clock at the very heart of bacterial evolution.
Now that wild reproduction speed becomes incredibly dangerous when harmless bacteria suddenly turn hostile. You see, the friendly normal strains in our gut, the commensal ones, they mostly just rely on what we call housekeeping genes. This is just basic DNA for routine, everyday survival, like cellular structure and energy. But the dangerous strains, they pack something called pathogenicity islands. Think of these as clustered genetic weapon caches, often imported entirely from other organisms. They give the bacteria the ability to stick to host cells, invade them, pump out toxins, and mess with our immune system. In a flash, they can turn an innocent microscopic bystander into a highly aggressive pathogen.
Moving right along to Section 2, the bacterial genetic arsenal.
So you might be wondering, how exactly do bacteria acquire these weapons in the first place? To get that, we need a quick anchor in human history. The biological breakthroughs that explain these superpowers were painstakingly pieced together by scientists over decades. From Frederick Griffith's early experiments way back in 1928, all the way to Watson and Crick mapping the DNA double helix in 1951, we've slowly uncovered the tools these bacteria use to survive. And let me tell you, they have quite a few tools.
The first major tool is the plasmid. The best way to think about a plasmid is like a genetic expansion pack or a cheat code. Bacteria have their main long strand of DNA, sure, but they also carry around these short, independent little rings of DNA. Plasmids harbor highly specific, super advantageous information, like the actual blueprints to produce enzymes that can destroy our antibiotics or literally alter the bacteria so a drug can't even bind to it.
And what's really wild is how they share these cheat codes. Because bacteria just reproduce by splitting in half, they can't mix their genes the way animals do. Instead, they use a process called conjugation. It's essentially bacterial sex. One organism will actually dock with another using this tiny, hair-like tube called a pilus. The genetic material, usually a plasmid, just flows right through this microscopic tunnel straight into the neighboring cell. It is the ultimate form of open-source sharing.
They don't just share, though, they scavenge. This goes right back to that 1928 experiment by the British bacteriologist Frederick Griffith. He discovered that if you took dead, highly lethal bacteria and injected them into mice alongside live, completely innocent bacteria, the mice still died. Why? Because those living, innocent bugs essentially acted like scavengers. They retrieved DNA fragments left behind by the dead pathogens and literally integrated them into their own genome. This process, called transformation, means even harmless E. coli can snatch powerful genetic weapons right out of their environment.
And get this, sometimes they even get a little unwitting help from viruses. It is super common for viruses, which we call phages, to infect bacteria. When a phage eventually leaves its bacterial host, it will often accidentally grab small bits of the bacteria's DNA on the way out. Then, when that virus goes on to infect its next bacterial victim, it shuttles those stolen genes straight into the new host. We call this transduction.
Now, inside the bacterium itself, things are just as dynamic. Back in 1941, the pioneering botanist Barbara McClintock discovered transposons. These are basically jumping genes. They are genetic elements that can literally physically move from one part of the chromosome to a completely different part. This internal genetic shuffling gives the organism incredible flexibility to adapt whenever its environment changes.
Because the encoded information isn't locked down in one fixed spot, it has a way better chance of actually being utilized.
Finally, bacteria are absolute masters of energy efficiency thanks to things called operons. Discovered by Francois Jacob and Jacques Monod, operons basically act as a physical cover for genes. Think about it. Instead of constantly wasting precious cellular energy cranking out every single enzyme it has the code for, the bacteria keeps certain genes shielded. The operon only pulls back the cover when that specific product is actually needed in the environment. It's an incredibly efficient just-in-time manufacturing system.
Which brings us to Section 3, the antibiotic resistance crisis. So E. coli and other bacteria clearly possess absolute mastery over their genetics. But here's the terrifying part. Those exact genetic superpowers are currently fueling a massive, global public health crisis. Let's look at the number 210. We have roughly 210 FDA-approved antibiotics in the United States. Now, that sounds like a massive, formidable arsenal for humanity, doesn't it? Well, it's actually a dangerous illusion. In reality, there are only a little over a dozen true, unique classes of antibiotics. We haven't discovered any completely new classes in years. The recent additions are mostly just minor chemical tweaks to the drugs we already have. Our well of new weapons is running dangerously dry.
Meanwhile, the bacteria, they're using their playbook to adapt. When Alexander Fleming famously discovered penicillin, he noted that it completely inhibited the growth of the bacterium Staphylococcus aureus. It was a miracle drug. But today, over 90% of the clinical isolates of that exact same organism are completely resistant to penicillin. Through genetic sharing and scavenging, they have evolved right past our best weapon. The real-world consequences of this are just devastating. We see it especially with gram-negative rods in MRSA, methicillin-resistant staph. When these highly resistant strains strike a patient, doctors are forced to resort to increasingly toxic backup antibiotics, and those drugs cause a lot more collateral damage to the patient's body. Worse still, the very first antibiotic a doctor gives based on their experience, the empirical treatment, it frequently just fails against these bugs. By the time the lab finally identifies a drug that actually works, a patient suffering from something like sepsis might already be past the point of no return. That leads us right into Section 4, testing and measuring the threat.
With the stakes literally life and death, how does the medical world fight back? Well, the process inside a clinical microbiology lab is surprisingly elegant and logical. It's called an antimicrobial susceptibility test. First, the scientists make various dilutions of an antibiotic. Next, they add an equal amount of the patient's infecting bacteria into every single dilution. Then, they just watch. They are hunting for the minimum inhibitory concentration, or the MIC. Simply put, this is the test tube with the absolute smallest amount of the drug where the bacteria completely fail to grow.
Once the lab pins down that MIC, they correlate it with the level of the drug that we can safely achieve inside a human bloodstream. If the drug can successfully stop the bacteria at those achievable blood levels, the lab grades the bacteria as susceptible, and they mark it with an S. But if hitting the MIC requires a drug concentration way higher than what's safe or even possible in the human body, the bacteria gets graded as resistant, marked with an R.
But there is a really crucial distinction here we have to talk about. The MIC, it only halts the growth of the bacteria. It basically just slows them down enough so the patient's own immune system can swoop in and finish the job. However, for some incredibly severe infections, just stopping the growth isn't going to cut it. We need to guarantee total, absolute bacterial death. In those cases, the lab has to determine the minimum bactericidal concentration, or MBC. They do this by transferring those clear samples over to an antibiotic-free media just to make 100% sure that absolutely nothing grows back.
Finally, let's wrap up with Section 5, stewardship and the ticking clock. We know by now that bacteria are just brilliant at adapting, but the harsh reality? We are inadvertently helping them rewrite their playbook. The term for responsible antibiotic use in medicine is stewardship, and frankly, we are really struggling with it. The main drivers of this entire resistance crisis are mostly human actions. Massive misuse in agriculture and industry has introduced non-human resistance genes straight into our ecosystem. Doctors sometimes cave and prescribe antibiotics for viral infections just to appease unhappy patients, even though we know antibiotics do absolutely nothing against a virus. People don't finish their prescriptions, they stash the leftovers in their medicine cabinets, and they try to self-prescribe later. And in some parts of the world, antibiotics are just easily bought on the black market. Every single one of these actions accelerates the bacteria's ability to learn, adapt, and share cheat codes.
And the scariest part is, we can watch this happening in real time. Hospitals mathematically track the effectiveness of their drugs using a tabulation called an antibiotogram.
It basically calculates the percentage of susceptible bacterial strains they've isolated over a given year. When you look at these numbers year after year, it is a slow, agonizing decline. An antibiotic that successfully killed 85% of E. coli strains one year might drop down to 83% the next year, and then down to 80%. The trend is totally unmistakable. The clock is absolutely ticking.
We are locked in a high-stakes biological arms race with an organism that can divide every 20 minutes, swap genetic cheat codes on the fly, scavenge for dead weapons, and constantly edit its own blueprints. So as these microscopic survivalists seamlessly adapt and share their genetic code with each other, we're left with one critical question to ponder. Will our scientific innovation and human stewardship actually be enough to outpace their relentless evolution? Thanks so much for joining me on this explainer, and keep learning!